Hello everyone, welcome to the week 10 PMRF-NPTEL lecture on PMRF-NPTEL session on Introduction to Developmental Biology. So for if you have been following the videos, you know that the PMRF-NPTEL sessions are designed in a way so that a PMRF fellow such as myself can go through the various problems and assignments that were assigned to the students taking the NPTEL course in that week so that we can interactively solve the various problems that are there and then understand how to exactly tackle the problems of this particular week and better understand the various concepts that were dealt in this week. So this is the week 10 lecture and we'll go through the content that we have done in the week 10 lecture momentarily and then go right through the assignments and the problems. First, let me introduce myself. My name is Anuvab Chatterjee. I'm a PMRF Fellow and PhD scholar under the supervision of Professor Jonaki Sen at the Brain Developmental Biology Lab, Department of Biological Sciences and Bioengineering, Indian Institute of Technology, Kanpur. So, in the week 10 lecture, in the week 10 lecture series, we had looked at plant development and further looked into how fluorescence or the process of how flowering occurs in angiosperms exactly happens and the molecular pathways of the same. We also went a little bit into mammalian development and looked at early mammalian development and as to how the process of blastocyst formation and blastoderm formation actually happens. And we have questions regarding those in this particular session. So if no one has any questions, we will go right into the into the assignment questions themselves. So let us get started. This is the first part, the first question that we will tackle today. The question is, which of the following was or were the outcomes of the analysis of mutant lines and clonal analysis in Arabidopsis? So in order to understand plant development, there were many analyses that were being done. One of the major ones were trying to knock out particular genes and trying to see if the particular plant develops properly or not. The main model organism for plant development was Arabidopsis. If you know about a bit about plant development, it is a very small flowering plant, and it is the mainstay in plant developmental studies. So, with the mutant analysis, what was exactly found? The options are that the shoot meristem is derived from the ground tissue. If you remember that there are three types of primary tissues present in plants: the ground, the vascular, and the dermal tissues. And so the first option is that shoot meristem is derived from the ground tissue. Second option is shoot and root meristems form independently of each other. Third is cotyledons originate from the hypophysis. And the fourth is cotyledons originate from the general embryonic tissue. So this is a more than one correct option because there are four. It's a round box type of question. So more than one will be correct. So anyone will try to answer this? At least guess which one could be correct. Okay. So let us go through the solution slide and try to answer this. So let us first look at cotyledons, what exactly those are. These are structures which aid nourishing by initiating photosynthesis before leaves form. So these are the first photosynthesizing structures present in developing embryo. And in some plants, they also serve as a food reserve. So here you can see the cotyledons out here in blue, these two parts in the heart-shaped embryo. Next is meristem. Meristem is essentially the counterpart of stem cells that are found in animals. And there are two kinds of meristems. One is the shoot apical and the root apical meristems that are formed during embryogenesis. And finally, the mutant studies in Arabidopsis, the mutants that were formed, such as shoot meristemless, hobbit, or topless, provide evidence that the two apical meristems are formed independently of each other. That is, the shoot apical meristem and the root apical meristem are independently formed. So the absence of one does not affect the other. Secondly, here we can see directly that The cotyledons are formed out of the blue tissue here, which will be part of the ground tissue and not the hypophysis. And thus, the options out of the four that are correct is shoot and root meristems are formed independently of each other, and cotyledons generate from general embryonic tissue and not just the outer part of it. So this entire structure is cotyledon. So that is that. So if anyone does not have any questions, we can move on. Okay. So the next question is, which of the following is or are true of the maize viviparous mutant? So viviparous is obviously a gene that was knocked down in the maize, and people try to understand what exactly was the effect of it. And the options are, in this mutant, the plant hormone gibberellin is produced inappropriately. So this is gibberellin is one of the hormones present in the plants. This is the first option. The second is these mutant plants fail to form root meristem. Third is these mutants are defective for seed dormancy. And the final one is in this mutant, the plant hormone abscisic acid is produced inappropriately. This is the only one type of correct option, and I would like you to try and answer this. So, which of the following are true for maize viviparous mutants? Okay. So let us look at the viviparous maize mutant, what exactly the loss of function of the gene tells us about the plant development. So this is one very important part towards the seed development or the embryo development in plants, is that once the apical basal polarity is established, or the as we say dorsal ventral, but in plants it is apical basal polarity is established in plant in the embryo itself, it does not immediately germinate to form the new organism. What happens is that the seeds need to be propagated far away from the parent plant in order to successfully germinate and form another new organism. So for that purpose, the embryo can't just directly go into producing the new organism or going further along the steps in development. What happens is that the seed enters a period of dormancy, in which once the basic body plan is there is a shift to creating food reserve and the development is stopped for the wild being. The genes encoding seed storage proteins are abundantly produced. So essentially, this is the food storage period in which the embryo stores food reserves around it and forms the seed. And this will be used by the embryo to develop into a new plant. So obviously, unlike the case of animals, the seed develops independently of the plant, and thus it needs an entire food reserve for itself while it develops. After the food accumulation process is over, metabolism slows down, connection to the ovary is cut, and the seed desiccates or loses water, and the integuments, that is the sporophytic part of the embryo, sporophytic part of the entire ovary chamber, harden into the seed coat. All the above process, that is entering into dormancy, is result of a precisely regulated genetic program. So the exact point where the seed should enter into dormancy and stop producing food is very precisely regulated. And when it should come out of the dormancy and start forming the embryo is also very much precisely regulated by the entire genetic program of the seed. The plant hormone abscisic acid is important for maintaining dormancy, whereas the gibberellins promote breaking dormancy. So while in dormancy, abscisic acid is more prominent, while the seed comes out of the dormant period, gibberellins are being produced. This is the viviparous maize mutant that was being talked about in the question as well. In this, as you can see, although these seeds are desiccated, are dormant, but these ones have started to germinate. Especially this one you can see right here. This is because the viviparous maize mutant is defective in dormancy. That is, it is not dormant as for as long as it is required to and starts germinating early. So this is the defect of the viviparous maize mutant. And which of the following are true in the maize viviparous mutant? So the option is these mutants are defective for seed dormancy. So does anyone wants to has any questions on this? I guess. So we move on. The next question is, when ingested by an animal, exposure to acid in its gut can contribute to a seed germination due to which of the following? So, as we just discussed, that the seed needs to be propagated far away from the parent plant in order to germinate and successfully give rise to a new organism. But how does exactly the seed know that it has traveled enough, or enough time has passed that it should start germinating now, or that the conditions around it are favorable for it to germinate? So there are various processes which determine this, and the seed, after going through a particularly harsh environment, develops this notion that it has propagated long. This harsh environment could be due to traveling along, be could be due to ingestion by an animal and exposure to acid in its gut, which will show that after the animal has traveled for a while, the seed exits its body and then can start forming a new organism. It is taken that the animal would have traveled and propagated the seed. So this also, there are other ways as well in which imbibition is one of the processes in which, as we have already discussed, the seed desiccates before going into dormancy. But imbibition is a process by which it retakes water. So once it starts retaking water, the entire genetic machinery starts coming back up out of dormancy, and the seed starts the process of germination. So these are the various processes that are required, but the question is, when ingested by an animal, exposure to the acid in its gut can contribute to seeds germination. And what is this process known as? The options are stratification, imbibition, rehydration, and scarification. This is one option correct, only because it's a round type of answer. So anyone will try to answer this? Sangeeta says it is A, stratification. Okay, so let us go ahead and look at what the terms exactly mean and try to answer this again. The environmental factors, temperature, water, light, and oxygen, are central to germination. And processes which are named here, right here, are the various processes that the seed needs to go through in order to understand that it now has to germinate. These processes include stratification, which is the requirement of chilling. So essentially, the seed, after going through stratification, knows that the period of winter has passed, and now spring will come where it is the environment around is okay for it to start germinating. Because in winter, probably it would not have, the environment would have been too harsh. So stratification is one of the process. Next is imbibition, which is the process, as I said, of rehydrating, where the seed starts taking up water again, and the primary embryonic root called the radicle aids in this process of imbibition. And finally, the process of scarification is the scratching of the tough seed coat, and this can happen when a animal ingests the food and the seed goes through the gut acids, and that produces scratching or roughness on its seed coat, and that is the process known as scarification. And after this process also, the seed understands that it has traveled enough away from the parent plant and can now go ahead and germinate. So the answer was scarification. Anyone has anything to add to this? If not, you can go ahead. So next, after looking at the seed development, we will now look at flower development in close detail. And the first thing we will look at is this question, which is what will be the consequence of CLAVATA1 and CLAVATA2 double mutation? So for context, this particular question comes from the angle of meristems and especially inflorescence meristems. In the normal plant, which is not at the moment flowering, most of the meristems go ahead and form leaves and branches and structures which are not at all related to reproduction, such as flowers. But when the change occurs in the plant that it now has to go ahead and form flowers, the special type of meristematic tissues, that is, this is the counterpart to stem cells in animals, start for going through a process of differentiation and forming the inflorescence meristems, which will, after further differentiation, give rise to flowers. CLAVATA1 and CLAVATA2 are genes that are related to the stem cell lineages, the meristem lineages, and they control particular aspect of stem cell differentiation and proliferation. So the question is, what will be the consequence of CLAVATA1 and CLAVATA2 double mutation if both of them are knocked out? The options are, this will cause an increase in meristem size. This will cause increase in Wuschel expression. Wuschel is another gene that is related to meristems. It will reduce the levels of Wuschel expression and it will produce extra organs. So anyone will try and answer this, which of these can be the correct option? Since this is a square box type of answer, so more than one is correct. Anyone will try and answer this? Okay. So let us first look at the gene circuit and how exactly Wuschel and CLAVATA1 and 2 and everyone is related to stem cells. So Wuschel and STM proteins keep meristem cells in an undifferentiated state. That is, so meristem cells can exist in two forms. One is the undifferentiated meristem state similar to what stem cells or heart, in which the meristems give rise to more meristems, and that makes sure that the entire meristem population remains constant is starting to increase. While the products of clavata genes such as CLV1, CLV2 and CLV3 limit the number of undifferentiated meristem cells. So how exactly CLV1, CLV2 and CLV3 do this? What they do is that they initiate differentiation in the meristem cells. So they allow the meristem cells to go ahead and differentiate to form various structures. And this reduces the number of undifferentiated meristem cells which could have formed various other structures as well. So this is essentially a stem cell to a normal somatic cell differentiation that is occurring here. And how exactly this happens is, clavata genes, as you can see here, such as CLV3 and CLV1 and 2, they inhibit Wuschel. And what Wuschel does is it helps to increase the stem cell population. So Wuschel tries to make sure that the stem cell population only proliferates, does not differentiate at all. Whereas clavata 1, 2 and 3 act together to inhibit Wuschel and hence cause the stem cell population to start differentiating into various somatic tissues. So this is what is occurring right here. And from this particular solution slide, we can try and understand the next one, in which we can see the effect of various clavata mutant phenotypes. So what exactly that, let us go back and understand what does clavata do. Clavata 1, 2 and 3 help in differentiation of stem cells. So if in the clavata mutant, this CLV1, 2 and 3 will not be there. And what exactly is the result of that? So is what we will see in this slide. So here we can see the wild type inflorescence meristems. Where we have this number of organs that we are being formed. These are all flowering buds. But in clavata 3-2 mutant, the inflorescence meristem is highly produces a large number of organs. compared to the A figure. This is because it's in the absence of clavata 3 and 2, if the stem cell population first proliferated a lot more before being allowed to differentiate. So more stem cells means more organs being formed, and thus we have a large number of flowering buds being present here. And this also translates into the flower that is being produced from these flowering buds. Ordinarily, the flowers we only have around 5 petals in the plants, and around I think 6 stamens are being formed. But in this particular plant, we can see 6 of these and a number of stamens being formed. And this shows that the clavata mutation, what it causes is it increases the number of organs in the final clavata mutant plant. Thus, the correct option is, what will the consequence of clavata 1 and 2 mutation? It will increase meristem size and produce extra organs. So we move on and to the next question, which is, what are intercalary meristems? So we have looked into what meristems are, and we have also looked into inflorescence meristems. We will now look into intercalary meristems. What are these? So the question asks us just that, and the options are meristems found in dicots, meristems that are responsible for secondary growth, the lateral meristems of monocots, or monocot meristems that are responsible for stem length increase. So these are the 4 options, and this is a round type of answer, so only one of them are correct. Anyone wants to try and answer this? Okay, so let's look exactly into what these intercalary meristems are. So the other 2 types of meristem that we have not discussed yet are lateral meristems and intercalary meristems. Lateral meristems are present, as you can see, if this is the along blown up part of the stem, and this is a cross section of it. The lateral meristems are present in this red line that is shown right here. These, and it is also shown right here, and what these do is that the lateral meristems help in increasing the girth or the width of the plants. On the other hand, we have intercalary meristems, which are present only in monocot stems, which are present only at the boundaries of, say, plants which have these internodes. So for example, you can look at them. So they are only present in the boundaries of these nodes, and here only this meristems help in elongation of the entire length of the stem. So they proliferate and produce more tissues, and this helps in the elongation along this part only. The middle part of the internode does not contribute to elongation, but only the intercalary meristems present at the edge of the internode. So intercalary meristems help in increase of the stem length size, and these are present only in the monocot stem. So the correct option is, what are intercalary meristems? They are monocot meristems that are responsible for stem length increase. So does anyone have anything to add to this? No. So next we move into another question on flowering of plants. Identify the correct order of steps involved in vegetative to reproductive transition in flowering plants. So what exactly are the steps which initiate the transition from the plant producing only leaves and branches to it producing flowers? So we have 6 options that we have to put into the correct chronological order and form the exact pathway in which the transition occurs from vegetative to reproductive phase. These options include, the first is CO, the gene CO activates the transcription of flowering locus T or the gene FT. FT interacts with the transcription factor FD. FT is translated to a protein. FD interacts with the protein to activate floral meristem and organ identity genes. CO responds to day length for promoting flowering plants, and Ft is transported to the shoot meristem. These are the six options, and we have to put them in the correct order according to the four options given right here. So let us go ahead and look into the vegetative to reproductive transition plants. So flower induction occurs by two main genes, which are constant tense, abbreviated as CO, and flowering locus T or FT. These are the main regulators of flowering induction. Further, constant tense or CO is responsive to environmental cues such as light and temperature around the plant themselves, which will signal the plant to actually go from the vegetative phase to the reproductive phase. Next is another other genes which include transition from vegetative meristem to an inflorescence meristem. These include terminal flower 1, which help the meristems to choose to change the identity from vegetative leaf and branch for producing meristems to flower producing meristems. And finally, we have floral meristem genes, which will actually give rise to floral genes. These include leafy, apetella 1, and cauliflower. So, what is exactly the interpathway that occurs? The first thing is CO or constant tense is gets affected by external stimulus such as light, and it is activated, and it goes ahead and activates the gene FT. FT is transcribed and it goes and activates various downstream genes such as TFL1, FD, and together with all of these genes give rise to a second order of floral meristem identity genes such as leafy and apetella, and finally leafy and apetella are the ones that play the major role in floral organ identity genes and formation of the actual flower. So the pathway is the external stimulus, constant tense, FT gene activation, FT transcript, FT activating FD, FD and FT together help in the activation of leafy and apetella, and and these finally give rise to the floral organ identity genes. So the correct option include uh, so identifying the order of or correct order of steps involved in the vegetative to reproductive transition in flowering plants. The first option is CO responding. The correct order is according to this particular chart. CO responds to day length and thus promoting flowering plants. That is will be the first. The 5 is the first. Second is CO activates transcription of flowering locus T, FT, as we just seen. FT is translated to a protein. That is the third. FT then goes ahead and interacts with transcription factor FD. FD interact the first will be the FT is transported to the shoot meristem, and then it interacts with transcription factor FD. FD activates interacts with a protein to activate floral meristem and organ identity genes, which include the leafy and apetal. So the correct option is 5, followed by 1, followed by 3 if it is transferred to protein, followed by 6 where FT is transferred to the shoot meristem, then 2 FT interacts with transcription factor FD, and finally 4 where FD interacts with the protein to activate floral meristem and organ identity genes. Though the correct option from these, it is number 2. So anyone wants to add anything to this? Okay. we move on to the following. So, according to the ABC model of flower development, what will be the consequence if B group genes are not expressed only in the third hole? So, let us first talk about the ABC model of flower development. So, we know that the flower has majorly four parts: the sepal, the petal, the stamen, and the carpel. What the ABC model of flower development tells us is that there are three categories of genes: the A, B, and C. The expression of these genes sequentially and in combination at a particular point gives rise to either of these four structures, that is sepal, petal, stamen, and carpel. Yeah, and this is what is described by the ABC model that we'll just see in the next slide. So the question is, according to the ABC model of flower development, what will be the consequence if the B group genes are not expressed only in the third hole? So there are four, obviously, corresponding to the sepal, petal, stamen, and carpel. And if the B group genes are not expressed, what happens to the third hole? So the options include: there are no abnormalities in flower development, petals will form in the third hole, the stamen will form in the third and fourth hole, and carpel will form in the third hole. There, it is an only one type of option, correct type of option, as this is a round type of answer. So, you will try and try answering this. Okay. So, let us first look at the ABC model itself. So, these are the organ identity genes that we have just discussed, which are activated after the activation of leafy and apetella 1. And I would like, so there are four classes, as I said, three classes, A, B, and C. The class A genes include AP2, which gives rise to sepal, combinatorial activation of A and B, such as AP3 and PI, gives rise to petals. Again, activation of B and C at a particular region gives rise to stamen, and only C gives rise to carpel. So I would like you to look at the middle diagram right here, and if we come at it from the outside in the wild type, at the biggest hole, only A class A genes are expressed, and so we have a sepal. Next we have A and B both being expressed, so we have a petal. In the third we have B and C being expressed, so we have a stamen. And in the final one we have only C being expressed, and thus we have a carpel. So depending on which one is expressed, we will get the corresponding structure. And this is the wild type, with the class A forming sepals on the outside, class A and B together forming petals, class B and C forming stamens, and finally class C forming carpels. And these are the mutants that we will just talk about now. So here we'll look at the wild type and mutant phenotypes of Arabidopsis class A, B, and C floral organ identity genes. And so what happens when we knock them out? So the first one we are going to talk here, which is directly related to the question itself: what happens if we knock out the B class of genes? So from this diagram particularly right here, we have A being expressed in the first one, so this will be a sepal, A and B here, which will give rise to petal, B and C here, which will give rise to a stamen, and C here only, which will give rise to a carpel. But in the B mutant, we do not have any B right here, and thus the structure looks such as these: A, C, and C. And we know A will produce sepal, and thus we have sepal in the first hole, again sepal in the second hole, and C we know alone will give rise to a carpel, so in the third also we have a carpel, and the fourth we again have a carpel. So the order changes from to sepal, sepal, carpel, and carpel, and these are the diagrams which show the actual plant. Similarly, if you look at the C mutant, So we do not have any C in the third or the fourth. Instead, we have expression of A only, and thus A alone gives rise to sepal, as in the first case. In the second one, it is also very similar to the wild type, where A and B are being expressed and forming the petals. So we have sepal and petal. But in the third one, where C should have been expressed, we again have A and B, and thus that gives rise to petal again. And in the final one, only A is expressed, which again gives rise to sepal. So we have sepal, petal, petal, and sepal, and no stamen and carpels are being formed. And this is the corresponding diagram. So you can see the petals right here, sepals which will be outside, then petals, and then again petals on the inside. So ABC genes are very, very important for a floral organ identity and gives rise to various structure in combination. Thus, according to the question, if we remove the B class genes, what happens is, what will the consequence if B group genes are not expressed in the third row? So in the third one, if B is not expressed, that is in this mutant, we have a carpel in the third row. So carpel will form in the third row. Anyone wants to add anything to this? Okay, so we build upon this information and go into the next question. Identify the correct statements regarding floral organ specification. The options are: in AP2 mutant plants, petals are made but sepals are absent. In AP3 and PI mutants, only sepals and carpels are present. In AG3 mutants, the innermost and the outermost floral organs are sepals, and the fourth class of floral organ identity genes is known as the sepatella. So out of these four, we have to choose which are correct. So this is a box type of answer, so more than one is correct. So will anyone try and answer this? Or try guessing which will be the correct option. No. So we'll go through the ABC model again and try to identify what is the problem. So wild type and mutant phenotypes of Arabidopsis class A, B, and C floral organ identity genes. So this is the wild type structure that we were looking at earlier. These are the four holes, 1, 2, 3, and 4. In the first one, only class A genes are expressed, which form the sepal. In the second, class A and B are expressed, which form the petal. In the third, B and C are present, which form the stamen. And finally, we have C only that produces carpel. So this is the wild type formation. Now let's look at what knocking out one class of genes does to the entire flower. So when AP2 or class A genes are knocked out, we have expression of class C throughout all the four holes. And thus, we have no A here but only C, and C gives rise to carpel, and we have formation of carpel. In the second, similarly, there is no A, only B and C, and B and C gives rise to stamen. In the third and the fourth, it remains similar to the wild type. B and C gives rise to stamen, and only C gives rise to carpel. And we have a flower forming like this, which does not have any sepals or petals. So if we can identify what the problem is, we're comparing it to the wild type. There is no covering to the entire plant, either from the sepals or the petals. Only the stamen and carpels are found. So we move into and look at these ones are the ones we just discussed. So in this one, we did knock out the B class genes, so no B on top of these. So we have the expression pattern of genes are A, C, and C. Because no B are here, so A will only give rise to sepals, and C will only give rise to carpels. And finally, if we knock out C, We have expression of A throughout, and A gives rise to sepal, A and B together give rise to petal. Similarly, A and B gives rise to petal, and A only gives rise to sepal. And these are the phenotypes that we will observe. But the story does not end here. It later results found that not only the A, B, C three classes of genes actually control the flowering of plants, there is a fourth one as well. These include the sepatella genes or ACP. So ACP1 and 2, 1, 2 and 3 are expressed in this manner, in which ACP1 and 2 are expressed throughout, ACP3 is only expressed in the final three holes. And all of these ABC class of genes act together with the sepatella genes in order to form the various structures. So to demonstrate this, if we knocked out sepatella 3 and sepatella 2 and still kept the class ABC genes intact, so we have A, B, C, C. The class genes are completely same as the wild type. But the sepatella genes are 2 and 3 are not there. So we have sepatella 1 throughout. We see the flower structure changes to all sepals. So even though the class A, B, C genes were not touched at all, we have change in the flower structure itself directly. So this gives us the notion that sepatella genes are very important for flowering or flowering structure formation as well. And these are the fourth class of organ identity genes. And here we have normal Arabidopsis leaf. And when we ectopically activate ACP in the Arabidopsis leaf, it forms a sepal-like structure. And thus the name as well, sepatella. Thus, if we go back to our question, let's look at it. So in AP3 and PI mutants, only sepals and carpels are present. In AP3 and PI mutants, only sepals and carpels are present, so this is true. In AG mutants, the innermost and outermost floral organs are sepal. So in AP3, AG mutants, sorry, in AG mutants. So in AG mutants, the outermost and the innermost are both sepals. And the fourth class of floral organ identity genes is sepatella. And in case of AP2 mutants, the in case of AP2 mutants, petals are made but sepals are absent. Sepals are absent is the option, but the actual one is that carpels and stamens are only formed. So the one is not correct. So the only ones that are true are 2, 3, and 4. So anyone wants to add anything to this? Okay. So we move on. So we have now moved on from plant development, and we will now move on to mammalian development and look at mammalian and try to understand mammalian embryo development from the ground up. So this is the first question that will be encountered here. That is, which of the following is or are characteristics of mammalian embryos that necessitate the use of other model organisms? So we have looked at various model organisms throughout the course. This includes Drosophila. We have looked at amphibians for a bit. You know that we also during development we look at chick, zebrafish. We have also looked a bit at plants, but we only look at mammals so late into the course, and mammals are also difficult to work with in case of development. But why is that the case? That is what the question is asking us. Which of the following characteristics necessitate use of other simpler organisms? We have also looked at C. elegans as well. So, what is the reasons for that? That is what the question is asking us. Is it the size of the embryo, the number of zygotes that are produced, early embryonic development is not accessible for studies, or asynchronous cell division? So, which of the following is the reason? Anyone will try to answer this? Okay. So we'll look at the early mammalian development and what exactly the causes that we look towards other model organisms for studying the development itself. The first reason is that the eggs are very small. There are very small eggs. The human zygote is about 100 microns in diameter, less than 1,000th the size of the frog. Next, the embryonic development happens entirely inside another organism, that is the mother, and thus the embryo is surrounded by mother's tissues, which are obviously we cannot look at the egg directly and look at it developing. In comparison to other organisms such as the frog, where the embryos develop outside the mother, there are very few zygotes that are present, and the cleavage zygotes are present that we have less number of opportunities to study. The cleavage is slow and rotational, and this is known as asynchronous cleavage. So, as we had seen that there is always no change in the plane of cleavage that occurs in frog embryos. That is, one is high, first one is horizontal, then meridional. This does not occur in mammalians, but there is rotational or asynchronous cell division. That is, the first plane of division can be separate to the second plane of division between as compared between cells. There is a process of compaction that occurs that again makes it difficult to study mammalian development. And there is another thing which is early activation of zygotic gene expression. So as we have seen in Drosophila development, that a major part of the axis specification is played by maternal effect genes, which are already deposited by the mother itself. But that is not the case in mammalian development. In mammalian development, the zygote itself is transcriptionally active very early in development. So the zygotic genes play a major role in various aspects of early embryo development. So the options that include which are the characteristics of mammalian embryos that necessitate the use of other model organisms. These include the size of the embryo, the small number of zygotes that are produced. Early embryonic development is not accessible for studies as the embryo is still inside the mother, and the asynchronous or rotational cell division, which makes it difficult to study. And thus, all of the options are correct. Anyone wants to add anything to this? Okay. So we move on to the next question. Which of the following is or are derived from both embryonic and extra-embryonic tissues? So during development, there is particular part of the embryo zygote which will give rise directly to the embryo, and there are parts that will give rise to extra-embryonic tissues, which includes the placenta or the connection between the mother and the developing embryo. So which of the following are derived from both embryonic and extra-embryonic tissues? These include the primitive streak, the embryonic mesoderm, the extra-embryonic mesoderm, or the amniotic ectoderm. So for this particular question, we'll directly move on to this schematic. This is a schematic diagram showing the derivation of tissues in human and rhesus monkey embryos. The dashed line indicates a possible dual origin of extra-embryonic mesoderm. So this is something that we will have to look into. So these are probably produced via lineage tracing experiments that we had momentarily seen way early into the course, in which we try to produce fate maps by looking at which by trying fate maps which will help us to understand which tissues are derived from which ones. So we have first have the blastocyst. This uh, bifurcates into the inner cell mass and the trophoblast. The inner cell mass gives rise to the hypoblast and extra-embryonic endoderm, which finally gives rise to the yolk sac, and epiblast, which forms majorly the embryonic tissues from via the embryonic epiblast, forming the embryonic ectoderm, the embryonic endoderm, and the embryonic mesoderm. These are the three layers that we have studied extensively throughout. And as you can see right here, the extra-embryonic mesoderm has origin from both the embryonic mesoderm lineage, so the embryonic lineage, as well as the extra-embryonic or this pink lineage, which are the extra-embryonic tissues. So there are these extra-embryonic mesoderm is derived from both of these, and thus the correct option for us here in this question, which of the following is are derived from both embryonic and extra-embryonic tissues. These are the extra-embryonic mesoderm cells. So we move on and look at the next question. With respect to molecular mechanisms leading to distinction between ICM and trophoblast, which of the following are true? So we have just seen that ICM is the inner cell mass and the trophoblast is this layer. So in the blastocyst, what happens is that after a process known as compaction, the cells in the interior of it form the inner cell mass, which will finally give rise to the embryonic tissues, as you can see right here, and the outer one will give rise to trophoblast. So what exactly is the molecular mechanism between which separates out the inner cell mass and the trophoblast is what we will look at into next. So what are the molecular mechanisms, and which are the following are true according to these? The first option is CDX2 protein is CDX2 is activated only in the ICM or the inner cell mass. CDX2 is activated only in the trophoblast. The mechanism is independent of spatial localization of cells, and the mechanism is dependent on the spatial localization of cells. So which of the four can be correct? It is a box type of question, so more than one is correct. Anyone will try to answer this? So we said the after the process known as compaction, the interior cells from the inner cell mass, the ones on the outside of the embryo from the trophoblast layer. So do you think we can choose between the options 3 and 4 because they are exactly opposite to each other? Only one will be correct. At least that we can choose. The mechanism is independent of spatial localization, or the mechanism is dependent on spatial localization. Which do you think will be correct? Here, spatial localization means the position of the cells themselves. Okay, so we'll look at solution slide itself. These are the proposed functions of the three transcription factors, NANOG, OCT4, and STAC3, in retaining the uncommitted pluripotent fate of embryonic mouse cells. So we'll look into that later. So at a stage known as morula, after the stage known as morula, the trophoblast and the ICM get separated out into two. The trophoblast, in the trophoblast, the genes CDX2 and EO mesoderm is activated. So CDX2 functions in order to inhibit the gene OCT4 and also the gene NANOG. And OCT4 helps in the formation of ICM, as well as STAT3 also helps in the formation of ICM. And from ICM, the formation of epiblast and embryonic tissues is helped by NANOG, whereas NANOG inhibits the formation of the hypoblast layer. So ICM, as we have just seen in the flowchart, gives rise to either epiblast or hypoblast. Depending on the presence of NANOG, it will give rise to epiblast or hypoblast. So looking at this particular diagram, we can see right here that trophoblast has CDX2 expression, and if we go right into this particular stained image, we see that OCT4, which is this transcription factor, which is helps in the formation of ICM, and thus the ICM cells will only express OCT4, is in the interior of the embryo, whereas hypoblast and trophoblast cells cover the entirety of the embryo towards the outside, which will probably be stained with proteins such as CDX2, stained for genes such as CDX2, and thus Uh, and thus, from this particular diagram, we know that trophoblast expresses CDX2, the first point. And second, the spatial localization of the cells is also very important because the cells in the interior are the only ones which will start expressing OCT4 and produce the inner cell mass. But the cells on the outside of the embryo will only produce the trophoblast cells. Thus, the spatial localization is also very important. Then this is a point that is we have to keep in mind, as we move on, that the how is it that the spatial localization plays a part. So how do the cells know that they are towards the exterior or the interior? This is something that we will look into in the next one. But for this particular question, with the respect to the molecular mechanism leading to the distinction between ICM and trophoblast, which of the following statements are true? The options are CDX2 is activated only in the trophoblast, and the mechanism is dependent on the spatial localization of the cells. These are the two options which are correct because according to how the flowchart that we have just seen. So anyone wants to add anything to this? No. So we move on and look into the mammalian embryo development. What is the role of hyposignaling at the morula stage? The options are: it promotes phosphorylation of YAP, it protects YAP from degradation, it targets LATs for degradation, and it facilitates the binding of TED4 protein to DNA. And so we'll look into it. So hyposignaling is the reason behind the spatial localization of cells, and or the spatial localization of cells plays a major role in hyposignaling, and hyposignaling in turn decides which cells will become the inner cell mass and which cells will become the trophoblasts. So how does this exactly happens? So cells which are towards the interior, which will give rise to the inner cell mass, are have cells all around them. So cells surround these cells completely, the inner cell mass cells. So such as this one towards the right. So it has cells towards every side of it. So hyposignaling, which is a close range signaling, or happens towards it from all sides, and thus hyposignaling is active in this particular cell. Hyposignaling, if it is active, it activates the protein LATs. LATs phosphorylates YAP, and phosphorylated YAP is degraded completely. So we have no YAP here. And thus there is another protein called TET4 that is bound to DNA, that is not very interesting here, but TET4 is a transcription factor for CDX2. And thus in this particular inner cell mass cell, CDX2 is not produced. That is how it should be, as we have just seen. But in case of a external cell which will give rise to trophoblast, we have that it is not covered by cells from all towards all ends. We have one end which always will be free, one or more. So hyposignaling is not active in this particular cell because it is not covered by cells all over, and thus LATS is not activated. If LATS is not activated, then YAP is not phosphorylated and degraded. If YAP is not degraded, it enters into the nucleus and binds to TET4 transcription factor, activates it, and helps in the production of CDX2. And thus, in the external cells such as the left one, no hyposignaling is there, YAP is not degraded, and CDX2 is produced. And as we have seen, CDX2 identifies the cells which will give rise to trophoblasts. And thus, these cells on the exterior will form trophoblasts, while these ones in the interior will not have any YAP, and TET4 will not go under allow the transcription of CDX2, and these will give rise to the inner cell mass. So the entire discussion of it gives rise to this particular question. So what is the role of hyposignaling at the stage of morula? It promotes phosphorylation of YAP. and which then goes ahead and helps in the transcription or not transcription of CDX2. Anyone has anything to add to this? Okay. So that was the entirety of hyposignaling that we discussed. And this is the next question on which is continuity of the, we continue on the previous one. So in mammalian embryo development, what will be the consequence of the absence of proteasome activity at the morula stage? So proteasome, for context, is the organelle that is responsible for degrading of proteins. So that is something that we have to keep in mind. And what is the consequence of absence of proteasome activity? The options are no CDX2 expression in any cells, no ICM formation, or the inner cell mass formation will not be there, no trophoectoderm formation, or CDX2 will be activated in all cells. This is a box type of answer, so more than one is correct. Will anyone try and answer this? So if proteasome activity is not there, that is proteins are not being degraded, which of the following will happen? No CDX2 expression in any cells, no ICM formation, no trophoectoderm formation, or CDX2 will be activated in all cells. Okay. So we'll look into the solution slide. We come back to a previous drawing schematic itself. As we have seen that hyposignaling gives rise to activates LATs, and this causes YAP to be phosphorylated and then degraded. This degradation of phosphorylated YAP is what is done by the proteasome. So if no proteasome is there, then YAP will not be degraded even though it is phosphorylated. And thus there will essentially be no difference between these two cells. YAP will be here as well as here. And then YAP, in spite of having active hyposignaling in this ICM cell, YAP will still go into the nucleus and bind with TET4, give rise to active CDX2, and thus in both the cells CDX2 will be there because in both of them YAP is not degraded. And thus if both of them express CDX2, both will form the trophoblast layer, and thus we will have no inner cell mass formation because everything will express CDX2 and everything that expresses CDX2 will form trophoblast, and thus we have no ICM formation and no degradation of YAP in these particular cells. And thus the correct option is, if in mammalian development, what will the consequence of the absence of proteasome activity at the morula stage? No ICM is formed and CDX2 will be activated in all cells. Is that okay? Okay. So that was everything that we had to discuss today. The references to the particular slides are the Introduction to Developmental Biology by Scott Gilbert, the 9th edition, and this is the YouTube channel link on which all the recorded lectures are uploaded. So if no one has anything to add, we can stop the session right here. Does anyone have any questions regarding the course material and the problems discussed this week? Probably not. So that is it for today. I thank everyone for joining, and I look forward to meeting everyone next week. Thank you.